With Doubtnut, get instant video solutions to all your maths, physics, chemistry, and biology doubts. Just click the image of the question, crop the question, and get instant video solution. Download Doubtnut app today.
Hello students, have a look at the question. Question is the optimum temperature for polymerization in PCR is blank, while the enzyme responsible for the mentioned step can tolerate temperature up to blank. Select the correct option according to the blank. So here we are giving the 4 different temperatures in different options. So firstly we will see PCR. PCR, that is polymerase chain reaction. It is polymerase chain reaction. This is a technique, this is a process which is used to amplify the DNA, or we can say that which is used to make millions of copies of DNA, that is polymerase chain reaction, copies of DNA. So if we'll see the steps of the polymerase chain reaction, it involves the 3 major steps. That is, first is the denaturation. 2 strands, they get denatured, they get separated. Denaturation, and second is the annealing means binding of the primers to the strand, annealing, and third is the elongation, or we can say that the extension, that is, or polymerization. It could be anything, extension, polymerization. And this is a denaturation. It occur at the temperature 94 degrees centigrade. 94 degrees centigrade. Annealing, it occur 48 to around 65. It depends upon the primer, that is primers used. It depends upon the annealing temperature of the primer, so that is on dependent on that. And extension, that occur at the 72 degrees centigrade. Now the enzyme that used is that used for polymerization. Definitely that should be the which can tolerate that much high temperature, because highest temperature in the denaturation is the 94 degree. So we can say that the enzyme that used is the tag DNA polymerase. Because in the while it's not like that if we are adding the enzyme, firstly denaturation step will done, then the denaturation, we have to add the enzyme and all the steps will be followed in a single while. So that means temperature, the enzyme should be the thermostable. So this enzyme, it is a thermostable means it can tolerate the high temperature, it will not get denatured at high temperature. Now our statement say optimum temperature for polymerization, extension, it is the same thing. So we can say that the polymerization is also same thing. The temperature is 72, that is 72. Here we can write that. Next is, while the enzyme responsible for mentioned step, that is enzyme responsible for polymerization is tag DNA polymerase, it can tolerate temperature up to, so it can tolerate temperature up to 95, because 94 already occurring in the vial, so it can tolerate up to 95 degree temperature. It is that much thermostable. So our answer is 72 and 95. Here is 95 wrong, 94 this is wrong, 72, 95. Polymerization temperature is 72, this is correct. 95 and 72, this is wrong. Correct is option C. Hope you get it. Thank you.
For class 6 to 12, IIT JEE and NEET level. Trusted by more than 5 crore students. Download Doubtnut app today.